Thank you very much. I'm Mahta Mehdiabadi. I'm a PhD student in the University of Padova. So today I will be talking about function prediction for disordered proteins and disordered regions using the graph autoencoder approach. First, I would like to give a quick overview of what disordered proteins are. So these proteins like a stable 3D shape, but instead they exist in a collection of conformations called ensembles. And a protein can be entirely disordered or it can be disordered in some regions. In that case, we call them intrinsic disordered regions. They might be short from 5 to 10 residue or span more than 1,000 residue in the protein. And they can be in the tails of the sequence or in the middle and act as a loop or a linker. And it's estimated that more than one-third of human proteome residues are disordered. So although these proteins lack a stable structure, they have key roles in cellular processes, and they usually act as hubs in protein-protein interaction networks, often via transient or multivalent interactions. And they have poor sequence conservation, and even in the regions that show stronger similarity in alignments and called short linear motifs and molecular recognition features, that have functional roles. And these are like a minor fraction of the residues, and they're not even positionally conserved. So we want to assign functional annotations to disordered regions. The first step to find these disordered regions. For that purpose, we use two databases. First is DISPROT, which is a gold standard database of intrinsically disordered proteins, but it has around 3,000 proteins, and that's not really enough for training a model. I will get back to DISPROT later, but that's why we use MobiDB. MobiDB is a aggregates disorder annotations from literature and from experimental evidence, along with predictions from disorder predictors. So here I have an example entry of MobiDB. And MobiDB provides us MobiDB Lite, which gets a majority consensus among 8 different disorder predictors. And as a consequence, MobiDB Lite is very conservative. It has few false positives. So if it says a sequence is disordered, we are sure that it is disordered. And it is also already integrated into InterProScan, and that's how we used it. But going back to DISPROT, well, it is manually curated. It relies on 3 different ontologies. IDPO, which describes the structural aspect of IDP and IDR self-functions and functions directly associated to disorder state. And then there is evidence and conclusion ontology, or ECOD, that describes the techniques or evidences associated with an annotation. And finally, the gene ontology. But DISPROT has a difference regarding gene ontology assignment with UniProt, and it assigns gene ontology terms to disordered regions instead of the whole protein. So here I have an example of a DISPROT entry. And as you can see here, it shows that it identifies the disorder regions and also assigns them a biological process and molecular function. And then these annotations of DISPROT is propagated to UniProt for the whole protein. And also in the bottom, okay. And in the bottom, you can see the evidence that supports these annotations. And here I have some statistics from DISPROT database. So I have the number of proteins, the disordered region, and the number of terms. And as you can see in the gene ontology, we have only 600 terms available in DISPROT. And this is a very small fraction compared with the 44,000 gene ontology terms. And this might be due to the fact that these disorder regions cannot do every function, but it's also due to the lack of experiments on these proteins. And so this motivated us to develop a method that can assign functional annotation to disorder proteins and regions without relying on a structure, because disorder proteins don't have a structure, and without multiple sequence alignment, because they have poor conservation in sequence, and do this in a scalable approach, because we want to do this for all the disorder proteins. And that's why we use a graph autoencoder that is a special type of neural network that handles graph data. And so basically we model each protein as a graph where nodes are disordered regions or domains. And they are linked together based on their positional approximity of them in the sequence. And basically this graph autoencoder lets us learn, reach, and compact embeddings of these nodes that are topologically aware of each other. So how we build a graph for a protein, we first use interpro to obtain domains and disordered regions by their starting and also their starting and ending in the sequence. We connect them together based on positional approximity of them in the sequence. And since we want to have some initial features for them to give as the input to the graph autoencoder, we use Proti5 embeddings, averaging them in the regions that they appear in the sequence. So to build our training set, we obtained the proteins that had domains and disorder regions from MobiDB and from Interpro, and that were in SwissProt. We removed overlapping domains and superfamilies. We removed proteins longer than 3,000 residue because of the limitations in computational resources. And we removed the sequences with more than 90% similarity. And in the end, we clustered these proteins based on their domains and the domains combination, because we didn't want repetitive graphs in our training set. In the end, we end up with 27,000 proteins. So this is an overview of the model. As an input, we give it the feature matrix, which is the Proti5 or ProTrans embeddings for the graph, plus the adjacency matrix of the graph. The encoder consists of a fully connected layer and then two graph attention layers. And the decoder basically is a simple matrix multiplication of the bottleneck of the encoder, and it tries to reconstruct the adjacency matrix of the original graph. We train this model in an end-to-end unsupervised manner, and it enables us to learn rich embeddings for each node for disorder regions and domains that are aware of each other. And for the inference, how we assign gene ontology terms to IDPs, we use the encoder. We encode all of our proteins that were modeled before as graphs, whether they had gene ontology terms or if they were in our query set. In order to get one embedding or one vector for each protein, we take the average of the nodes. So we end up with one embedding of 64 dimension. And then we do a KNN search against the proteins that we know already their gene ontologies, and calculate a weighted score, a weighted average score for each gene ontology term. And to test this method, we use this broad database that as I said, it is a gold standard database. We built two data sets out of it, one fully disordered and one partial disordered. And here I have the results of the model. So our model, and here is a comparison with BLAST, but this is not a naive BLAST, actually. It assigns the heat, like it's different from the CAF and the BLAST baseline that is used in CAFA setting. And because in CAFA, I think they assign identity score, but here we transfer the annotations with a score of 1 to our query protein for each hits that are found for it. And so we have like more F-score than BLAST in molecular function and cellular components. We have higher recall in all the ontologies. This is for a fully disordered set, and on partially disordered set, we have like higher results. It's obviously easier to predict these scores because they're a combination of domain and disorder. And for assigning gene ontology terms to disordered regions, here instead of searching the protein against all the gene proteins that we already know their gene ontology, we take the disorder node and we search it against the disparate database that we already know the gene ontology of the regions. And again, we calculate a weighted score for each gene ontology for each region. But here we have a problem, and it's that for us to be able to transfer functions, first, MobiDB Lite has to be able to find that predicted disordered region. And there are not so many overlaps between MobiDB Lite and Disprot, so we could only calculate the scores for molecular function, which we end up with having 31 proteins in the data set. So we have a F score of 0.54, a precision of 0.55, and a recall of 0.53. And for concluding remarks, we aim to assign gene ontology terms to IDPs and IDRs using a graph autoencoder. And while BLAST in some cases shows high precision, but it has low coverage and it doesn't have a high recall in most of the sub-ontologies. And for future work, we will be predicting functions for disorder regions, and they will be available in the MobiDB database. And I would like to thank you for your attention, and also thank my supervisors, and Damiano Piovesan and Silvia Tosato.
Okay, that was a great talk, and we're slowly segueing from biophysics and structure into the combination with machine learning. So I'm really excited about this type of work. We have some time for questions, so maybe I'll open. So my question is, as someone who's been frustrated, not that frustrated, the other frustrated, working with prediction of function of transcription factors, they tend to have two business bits. One which is well ordered that binds the DNA. That's not our business here. The other is the not well ordered that binds whatever it has to bind. And then drives it to the DNA and binds it there. And I'm kind of curious if you can expand a little bit on how you are assigning Go terms to, and how specific you can get with Go terms assigned to disordered regions. So Go terms are really Go terms are called PF terms. They're really protein function terms. Mm-hmm. So, and how specific, how good is your prediction there?
I mean, I don't have the results for specific terms, but for now, since we search for now, since our search space is Disprot database, if that function is in Disprot, like already a disorder region and annotated it, with it, we are probably able to find it. But the problem is that Disprot is not that complete, and we have to find a way to kind of expand the search space that we have, and that's what we're currently doing.
And quick other question is that when you assign the function, you assign it to a set of residues, like to a domain, or the
yes,
entire
to the predicted disorder region.
Got you. Okay. All right. Thanks.
Yes.
Hello. Hello. Can you hear me?
Yes.
Okay. So, sorry. So, in the training pipeline, you mentioned that you have set a sequence similarity of 90%.
Yes.
Can you elaborate more on why 90%? Why not 50, 60, or something else?
Yes. Well, 90% because we didn't want like 90%. I think it's a threshold that is mostly used to remove sequence similarity, but our main point was actually the last one, removing domain combination redundancy, because we build graphs, so we don't really care about the sequence at that level, but we care about the combination of domains that are appear in each graph. So we don't want the graphs of similar structure like a lot of times in the sequence, in the training set.
Okay, I get it. Thank you.
So do you exclude the Go term protein binding?
Sorry?
So there's a Go term called protein binding,
Yes.
which I assume has a great, it dominates the disorder prediction.
Yes.
Because many disorder protein binding. So do you exclude that specific goal term?
No, no, we don't, because a lot of these disorder regions, as you said, have that binding and
Yeah, so I think
excluding that.
it is okay to include its child term, but
Okay.
that goal term in particular.
Yeah, well, actually, a lot of disorder proteins are like have shallow annotations. So by removing it, we would end up with only the root of the ontology. So it's not that.